Well, I'd like to welcome you. Good morning, and this is the CurePSP West Coast Conference on PSP, CBD, and related brain diseases. My name is Richard Gordon Zyne, and I am the president and CEO of CurePSP, and I've been in this wonderful position since 2004 and have really enjoyed my work helping folks with PSP and the families and caregivers, and helping to raise money for research. It's truly been a wonderful experience to work with CurePSP and to acknowledge its 21 years of service to patients and families managing with neurodegenerative brain diseases. First of all, I would like to thank the Peebler PSP Research Foundation for its continued generosity in support of our research program, and all of our generous donors, big and small, who continue to support our critical mission programs and services. And my purpose here this morning is to give you an overview of CurePSP, and in particular, our research program. I'm going to talk because I've got a long presentation, so, okay. CurePSP has a vision of a world free of PSP, CBD, and related brain diseases. That is our ultimate hope, and I firmly believe that sometime in the not-too-distant future, we will have a good understanding of these serious diseases, and perhaps there will be treatments to halt them or even prevent them altogether. And I am optimistic that there will certainly be improved treatments to manage symptoms and even drugs to stop the destruction of brain cells. Our vision of a world free of PSP and CBD is based on hope, because hope matters. CurePSP is the only organization in the United States and Canada that focuses its resources primarily on PSP and atypical Parkinsonisms. These disorders resemble Parkinson's disease, especially with regard to many of their symptoms. But deep in the brain, they may actually be closer to Alzheimer's disease and other frontotemporal dementias in terms of how they affect the brain. In addition to progressive supranuclear palsy and CBD, corticobasal degeneration, the Foundation also supports patients and caregivers who struggle with MSA, multiple system atrophy, and a handful of other extremely rare brain diseases. All of these disorders are extremely complex. They often confound neurologists and healthcare workers, and as we all know, their symptoms can be devastating to patients and their families. CurePSP's mission is laser-focused on understanding these diseases so that we may eventually manage them, or better yet, eliminate them entirely. CurePSP has a service program aimed first and foremost at finding a cure or prevention of these diseases through research. But research is complemented with a program of support for patients and their caregivers. Since understanding and diagnosis and treatment of PSP and CBD are done by trained healthcare professionals, CurePSP is continuing to develop a program of education for neurologists, nurses, physical therapists, occupational therapists, and other helping professionals. We know that there are still many medical professionals who need to be properly informed about these atypical Parkinsonisms so that they may make more informed diagnoses and offer more appropriate treatments. CurePSP is the primary patient advocacy organization for people with PSP and atypical Parkinsonisms. We need to enlighten lawmakers and government officials about our diseases so that they may make informed decisions about funding for research and public support for patients. Most of them have never heard of these disorders, and it's important for them to understand the impact of these diseases on families. CurePSP works with the Parkinson's Action Network and the National Organization of Rare Disorders to help us in our advocacy work. Research goals. People are always asking me about research. How come you haven't found some sort of a cure yet? You've been funding research projects for 10 years, 15 years. What benefits have come out of your research? Why aren't you promoting the such and such diet? It helped my mother. What about well water? What about heavy metals? My father was a miner and he now has PSP. I lived on a farm for 30 years, and I think I got PSP from all the pesticides that we used to spray on the plants. Do you use stem cells in research? If not, why don't you do it? It can't hurt. Or there's this doctor in China or Israel who has been transplanting healthy brain cells into sick brains since 1995, and he's had great success. Yes, there's an urgency in research, and every person who is suffering from PSP and CBD deserves a good answer. And I speak to many people, and I will speak to anyone who calls me about their need or suggestions, but I often cannot give the answers that provide solace or relief. I can only present science and try to be a source of comfort and hope. Good research is based on solid science. It takes nothing for granted, assumes nothing at the outset, and must be objective. And what is learned must always be shared with other scientists. CurePSP's research program, under the leadership of Lawrence Golby, has served as an incubator for innovative projects that have served as foundational work for clinical and therapeutic interventions. We promote research that helps generate an integrated understanding of PSP and related diseases at both the molecular and the cellular level. And to this end, CurePSP provides grant support for the highest quality and most promising research. And it disseminates the results of the research and provides a forum for collaboration among researchers. And since 1997, the Foundation has awarded over 125 grants valued at over $9 million. Now, $9 million may sound good, but it's really a very small amount when you compare it to what has been raised for other major diseases over the years. The research program has 4 basic goals, all of which are worked on concurrently. At the same time as we are trying to understand how these diseases originate, why they start in the first place, and how they affect the brain and the body, we're also trying to identify which people get them, the genetic components, and how the environment might also be a factor. Because it's always better to recognize and stop a disease in its infancy, CurePSP wants to help develop diagnostic tests and biomarkers so that we might be able to see those diseases in action at an early stage and perhaps nip the disease in the bud before it blossoms into full-blown PSP. So our research desperately wants to develop a blood test or a spinal fluid test or an X-ray or a CT scan to be able to see PSP when it's just starting out. Doing research is like looking for a lost child in a giant forest, but our forest is immense. Think about trying to find a lost child in a forest the size of the United States. He could be anywhere, in the mountains, on a river, in a desert. And believe me, if our scientists knew exactly where that child was located, they'd go to him directly. So we have to begin at a point in this giant wilderness and simply identify the disease by what it appears to do to the body, such as the symptoms. And once they've given it a description and a name, they begin to search and move in various directions, just like searchers looking for the child in the forest. And back in 1964, doctors Steele, Richardson, and Olszewski did just that and gave a name to progressive supranuclear palsy, PSP. Sometimes the search is for those very basic causes of the disease. Maybe it's genes, maybe it's the environment, maybe it's trauma or previous injury, or maybe it's a combination of these factors. And at the same time as we look for causes, we're searching for ways to help people while they are suffering, and looking for ways to detect the disease when it can't be detected by our usual tests. And then there is research to help people to live better, to improve their quality of life by developing palliative treatments of symptoms, to help people see better, to be less depressed, to have more energy, to be steadier on their feet, and possibly even to prolong life. And of course, we're all looking for the magic pill, the drug that will completely halt the disease in its tracks or even prevent it from happening. The clinical trial is a vital part of this research. Sometimes we're successful, but other times the medications either don't work or are not tolerated by patients. And while all these lines of research are going on, the scientists are sharing their findings. Think of the searchers in the woods who find the footprint of a child or perhaps the sneaker as they walk down their line of research. All of these findings are published and shared. CurePSP funds research through 5 general areas. First one is investigator-initiated research projects, and these come directly from the scientists. These are researchers who are clinicians, come to us with their concepts and their ideas to study a specific part of the puzzle. It might be an aspect of genetics, a study of one of the various proteins that affect the cell degeneration, an exploration of the workings of a part of a cell, an epidemiological study, or even a treatment-oriented project. Most of CurePSP's research through the years has been done in this manner, and we have learned a great deal through these projects. Recently, there has been a tremendous effort in the area of genetics, and since 2007, CurePSP has been the primary source of genetics research in the world for PSP. The Charles D. Peebler Genetics Program has been instrumental in exploring some new genetic components and aspects for PSP and CBD. The CurePSP Brain Bank at the Mayo Clinic has been instrumental in doing research on brain pathology and is the prime source of brain tissue for the Peebler Genetics Program. Our research, especially our genetics, would be impossible without this invaluable resource. Our scientists are always publishing their research and sharing the data with the world. Our annual research symposium enables those scientists who are currently receiving funding from CurePSP to make presentations to our scientific advisory board and to share their ideas and accomplishments with scientists from around the world. Well, what have we learned? And we've learned quite a bit. Over the course of a decade, our research has taught us that PSP and CBD are under-recognized, underdiagnosed brain diseases and are often misdiagnosed as Parkinson's disease because of slowness of movement and balance, or misdiagnosed as Alzheimer's or even depression because of changes in mood, intellect, and personality. The root causes of PSP and CBD are still unknown, but there may be both a genetic component and an environmental toxic component for these disorders. Our research also holds insight into understanding other types of atypical Parkinsonisms, as well as Parkinson's disease itself, as well as Alzheimer's disease. Understanding brain proteins is essential to understanding neurodegeneration. A variant of the tau gene is more common in persons with PSP than it is in the general population. This gene produces a protein found to abnormally clump in brain cells. So understanding how neurofibrillary tangles develop in PSP and CBD has emerged. Although these tangles are found in other brain diseases, specific work on the tau gene indicates that PSP tangles differ from those found in other diseases such as Alzheimer's. Alteration in cell energy metabolism occurs in PSP. This alteration is also found in cells located outside the brains of PSP patients. This suggests that molecular level changes associated with PSP may not be limited to the brain. Such changes in energy metabolism could also result in oxidative stress, a process now known to occur in brain cells of PSP patients. Various laboratory animals have been genetically engineered to develop PSP-like changes in the brain. These animal models greatly accelerate the search for treatments for PSP in humans. And genes other than the tau gene also contribute to PSP and point to other areas in the human genome where such genes may reside. And that takes me to the genetics program. While genetics may not hold all of the answers to PSP or CBD, an understanding of the role that genes play is critical, and that's why we've spent so much effort and time and resources in this area. We feel that there is an important interplay of genetics, molecular biology, environment, and toxins in the development of these diseases. Technology is now available that permits fast testing of individual brain tissue samples for thousands of genetic markers covering every known gene. This technique involves scanning DNA from a large number of patients. This powerful technology forms the foundation of the WGA, or whole genome analysis, which has just been completed by our genetics consortium under the leadership of Gerard Schellenberg at the University of Pennsylvania. So completion of the WGA is only the first step in the work of the genetics program. And subsequent phases will study biochemical pathways and various proteins with the implicated genes. Well, what have we learned so far in genetics? Well, very soon the CurePSP Genetics Consortium will have its findings published in a major scientific journal, and the Foundation will conduct a major public relations effort to promote the study and spread the message about our need to cure PSP. The Peebler Genetics Program completed the WGA with over 2,000 patient samples with PSP and CBD and over 6,800 controls, people without PSP. And the result was 6 gene variants that were much more common in the group with PSP or CBD. First, the genes that have been newly incriminated in the cause of PSP and CBD affect the production of tau protein. The next step is to determine just what this gene is doing and look for ways to fix it. Our scientists think that the newly found variant simply increases the amount of tau protein. And if that's confirmed, then clearing excess tau from cells could slow or even halt the progression of PSP and CBD. Second is the effect on vesicles, which are like tiny water balloons used for transporting chemicals around the brain. They're critical, and if they malfunction, the cells will die. These are 2 defects, genetic defects that help the brain package chemicals inside these vesicles. We need to find ways of compensating for these genetic defects. And a first likely step would be to remove one or both of these genes from an animal model, observe the results, and then attempt to correct it with drugs or gene therapy. Third is the gene defect on myelin, which is the insulating material around the long tubes called axons that grow between brain cells and conduct electric current. The axons also transport vesicles from one part of the cell to another. And while loss of myelin is not known to be a feature of PSP, a defect in myelin could impair the function of brain cells. Fourth, and perhaps most important, are the 2 gene variants that impair the brain's ability to dispose of misfolded proteins. Such a malfunction is strongly suspected in Alzheimer's, Parkinson's, and other neurodegenerative brain diseases, and the cause remains unclear. In PSP and CBD, the misfolded proteins, if not properly disposed of, can be toxic and kill brain cells. We have already have ways of improving brain cells handling of misfolded proteins in laboratory animals, and will attempt to adapt techniques for practical use in humans. And fifth, another advance provided by the genetics program is a diagnostic test for PSP and CBD that does not have to wait for persons to develop symptoms. Conclusions. First, we are closer to understanding how genetics affects the development of PSP, but there's a lot more research to be done. Second, while there is probably a strong genetic component to PSP and CBD and related atypical Parkinsonisms, the effect of toxins and trauma and other environmental triggers is still not known and must continue to be researched. We hope that the NIH CurePSP-funded epidemiological study currently underway by Dr. Irene Litvan will provide some salient results. Third, there is hope that new pharmaceuticals now currently under study in clinical trials may help to alleviate symptoms and improve the quality of life. And today, Dr. Boxer will be discussing his clinical trials with the drug davunitide. Fourth, there is hope that continued treatment of neurological symptoms will improve patients' abilities to maintain a higher quality of life. Dr. Bordelon will share some of her treatments and trials. As I've said before, there is hope that a cure or prevention may be reached during the next decade. But there must be a sense of urgency in this country to the needs of people with serious neurodegenerative brain diseases such as PSP and CBD, and we must find that lost child in the forest. CurePSP is determined to see its vision of a world free of PSP and CBD come to fruition, and we must all work together to find a cure. I thank you very much, and I'm not going to take any questions. We're going to get together as a panel after Dr. Bordelon and Dr. Boxer give their presentations. So what I would like to do now is to introduce our next speaker. If I may, let's see here. Is Dr. Bordelon? Okay. There you are. Okay, there's Yvette. Okay. Dr. Yvette Bordelon received her MD and PhD degrees from the University of Pennsylvania School of Medicine in Philadelphia. She completed residency training in neurology at the Massachusetts General and Brigham and Women's Hospitals, and then a movement disorders fellowship at Columbia University. Dr. Bordelon is assistant professor in residence in the neurology department at UCLA. Her clinical work involves the diagnosis and treatment of Parkinson's disease, Huntington's disease, and other movement disorders. Dr. Bordelon also plays an active role in studies and clinical trials on neurodegenerative diseases, and she's also a member of the CurePSP Board of Directors, a member of our research committee. Dr. Bordelon.
Thank you very much. Thanks very much for the introduction. Thank you to CurePSP, not only for holding the